sorry. So the Theraphosity is composed by 107 genera and almost 1,000 species, and within Theraphosina there are classified 14 subfamilies. The most diverse of them is Theraphosine with 65 genera and more than 500 species, and the representatives of this subfamily are well differentiated by the presence of an extended subtegulum, very permanent kills in the embolus, and the presence of urticantine type 1, 3, and 4 in the abdomen, and the geographical distribution is limited to America. So in the last 30 years, the taxonomy of tarantulas has been described as chaotic, and this is because many currently valid taxa were described in late 18th and are diagnosed based on a few characters. And the descriptions are based on on a few specimens. Some of them are not completely developed, and also they lack specific localities. And this information is very important because it gives us an idea of their geographical distribution and also the range of the species. Also, as Caroline mentioned yesterday, in the last years, because of the expansion of tarantulas in market, a few species has been described from based on hobby material and also on exuvia. Most of the taxonomic revisions are based only on morphology, and recent studies incorporating molecular data have proved that some morphological characters have extended patterns of homoplasy. So one of the most recent studies incorporating molecular data in the study of tarantulas is that one of developed by Turner and collaborators in 2017. And they reconstructed history of the gene 16S to prove the monopoly of Theraphosine. And they have found that Theraphosine within Theraphosine recovered 3 main clades that they propose them to be considered as tribes. So we have the tribe Theraphosine, Grammostoline, and Apalopine. And the members of Grammostoline are limited to South America, but the members of Theraphosine and Apalopine are well extended in all the continents. So we have some species that are well known, as those of genus Brachypelma Theraphosine and Grammostoline. And in the tribe Apalopine, there are dwarf and medium-sized tarantulas that usually show a dorsal coloration pattern of the body. So within the tribe Apalopine, there are 15 genera and almost 100 species, and one of the representatives is the tarantulas of genus Davus. And these are commonly known as tiger-rump tarantulas because of the presence of transversal orange and black lines on the abdomen. The last revision of the genus was conducted by Gabriel in 2016. And after his revision, he found that the genus Davus is composed by 4 species. Davus pentaloris distributed from southern Mexico to Nicaragua, Davus ruficeps in Nicaragua and Costa Rica, Davus fasciatus from Costa Rica and Panama, and Davus santus known only for a locality in Panama. And this classification was based mainly on the dorsal coloration patterns of the body of the species, but also he revised the morphology as sexual structures of males and females. So according to his revision, Davus pentaloris has a widespread distribution, but also he found high morphological variation through this geographical distribution. However, many recent studies focused on megalomorph have found that they usually have conservative morphology and limited dispersion capability with some exceptions. And this is because of their sedentary habits. And this limited dispersion capability usually cause local endemism and high genetic divergence. So this has raised the question if there is underestimated diversity in many groups of megalomorphs. So Davus pentaloris was originally described from Guatemala. However, when I started revising the collection of the natural collection of arachnids of Institute of Biology of UNAM, I found many specimens of different populations and localities in Mexico. And I found very huge variation, not only in the dorsal coloration pattern, but also the male sexual structures. And for tarantulas, the sexual structures of the male are very important characters to separate the species. So all this variation raises the question if there is only one species in this group. So the research questions that have arisen in this study is that dual population in Davus pentaloris has been recorded from southern Mexico and Central America belong to a single species widely distributed and with considerable morphological variation, or is this morphological variation showing us that there is more than one species? So I revised many specimens deposited in national and international collections. I found 88 specimens from 33 localities, and 32 of these localities represent new records. So the material is from the states of Guerrero, Oaxaca, Veracruz, and Chiapas in Mexico, and also I included some samples of Guatemala and El Salvador. For the morphological revision, I observed specimens and structures on a stereoscope in order to find some characters that could indicate us that there are more than one species. Also, we took several photographies and measurements of many parts of the body to compare them between the specimens of the different populations. For the sexual structures, I followed Bertani of 2000. However, I made some modifications in the names of the structures of the bulbs palpal bulbs, and the measurements were following Hamilton and collaborators in 2016. So, because we don't only want to include morphological information, and also we want to explore the variation within Davus pentaloris, we included molecular data, partial segment of cytochrome oxidase subunit 1 of 963 base pair. All the molecular protocols were conducted in the National Laboratory of Biodiversity and at the Institute of Biology at UNAM. So for species delimitation, we use the general lineage concept of species. We use 5 species delimitation methods based on morphological and molecular evidence that were considered as secondary criteria. And the species delimitation was based on the congruence of the different evidence. So we use the morphological characters found in this study, and also for the molecular data, we include 2 methods based on genetic distances and 2 methods based on trees and coherence theory. And at the end, we compare the different results obtained with these methods and this evidence, and we arrived at an iterative solution to propose species hypotheses. So for the morphological approach, at the end we only revised 52 specimens because many of them were juveniles or subadults, and 28 of them were males and 22 of them were females. And we found that some qualitative data were important to separate these morphospecies. We found 8 somatic and 22 sexual characters that could help us to separate these morphospecies. And also we obtained 64 quantitative characters. And also we took more than 350 photographies. So this is one of the characters that could help us to separate the morphospecies in Davus pentaloris species complex. So the dark markings in the carapace, the coloration of subsegments of the legs, like some morphospecies have stripes in the patella, and also the morphology of the male palpal bulb, in like the presence of the different keels, the shape of the base of the embolus, and also the shape of the projection, the posterior projection of the embolum, and also because they have some tiny projections at the tip. Also, we use the morphology of tibial apophysis and the shape of the tibia 1 and palpal tibia. and also the dorsal and ventral coloration of abdomen of the different individuals. So we revised the females, we found that yes, the dorsal coloration pattern is very indicative of the different morphospecies. However, when we revised the sexual structures, we found very high interpopulation variation in the spermatheca. So, for example, the first six images represent different females of the same morphospecies. And the numbers below these images correspond to the length and width of the carapace. So we found, yes, that there is a transformation from two partially fused receptacles to one completely fused receptacles. However, this is no linear because we found very huge females that have intermediate states, and also small females that had almost completely fused spermatheca. So we are exploring this variation. At this moment, we're maintaining some females in captivity just to see the transformation of the spermatheca when they are molting, because yes, there is the transformation between one when they are very tiny to when they are growing is very marked. So we consider that the spermatheca is not a reliable character to establish species limits. However, in the case of males, we found that yes, they are very important characters to separate morphospecies. However, we could not incorporate morphological evidence of three populations because we only had females and juveniles. So, considering the available data, we identified 13 different morphospecies, and these are separated by the dorsal coloration patterns, but also the morphology of the male palpal bulbs and the tibial apophyses. Because from some populations we have only a few specimens, we could not apply morphometrics. However, the quantitative data was used as additional information to support the species hypothesis. For example, the total length of the body was very useful, especially to separate morphospecies that were closely distributed. For example, this from Paso de Milpa, Veracruz, in the adults males, the total length was about 33 millimeters, and a species that is closely distributed, geographical distributed, the total length is about 22 millimeters. And also, we found other characteristics that could help us to separate these morphospecies and to be useful in the taxonomic revision. Because, for example, the difference of the segments of the leg 4, like the comparison between the femur and metatarsus, was very informative. For example, in almost all morphospecies, the metatarsus was longer than femur. However, in some morphospecies that were very closely geographically distributed, this character is reversible. So the femur is longer than the metatarsus, and we have shown in other species of other genera that this character is it not varies in this in the length, and in the that to be some individuals have a shorter or longer femur. No, this is a very constant character, so it is useful to separate these morphospecies. For the genetic approach, the genetic distances that were calculated, we use them into two methods, and they recover 16 molecular taxonomic units. And the intraspecific distances between in these taxonomic units was about 0 to 1.2%, and the interspecific distances were about 2% to 12.9%. And they recovered three main clades in neighbor-joining. And these clades are separated by distance of more than 10% of genetic distances. So this is the core gene tree. And as you see, this is the 16 molecular taxonomic units. So also because we want to see what's happening with tarantulas, with these tarantulas about the trademark, we include 2 sequences of specimens of pet trait to see if they are recovering from different populations or they belong to the same population. One of them was obtained from material of Europe, and one of them from material from Mexico. And both of them were recovering the same molecular taxonomic unit. So it's possible that this population is in risk because of illegal trade markets. And also, when we mapped some characters that were useful to separate morphospecies, we found that some of them could be very indicative and could be putative. I mean, could be some characters that could be characterizing some clades. But however, we need to do the analysis, the classic analysis to prove if there are if these hypotheses are valid. So we found, for example, that some morphospecies have the legs with lack of only one species was found to have legs with lack of coloration. However, the dark marks in cap brands are present in several morphospecies. For example, in many species of this clade, there is a presence of retrolateral kill of embolus. No? And so this could be very informative that the morphology is very useful to separate these morphospecies, and also it proved also or corroborate this hypothesis obtained with molecular data. So this is the summary of species delimitation methods and integrative solution. This is the main bubble groups of the population that we have information. The blank rectangles are those populations in which we could not obtain morphological data. And this is the different results of the different methods used with molecular data. And so we obtained 13 confirmed candidate species. This is because these species were confirmed by morphological and molecular evidence. And we include Davus pentaloris, that is here. So the rest of the confirmed candidate species represent new species. All of them are from Mexico. And we recovered 3 unconfirmed candidate species, 2 from Mexico and 1 from El Salvador. So we need to incorporate the morphological information to prove if the morphological evidence is congruent with the molecular data.
You have 3 minutes to finish.
Yes. So this work has been published a few months ago. So if you want to read it, I can share the PDFs. You can write me an email and I send you. And also we found that the different morphospecies or candidate species are congruent with the geographical distribution. We need to also test some analysis of paleography to see what's happening here, and also because we need to incorporate additional specimens of different populations that had not been included in this analysis because we could not obtain molecular data. However, it's very interesting because 2 of the species were sympatric. However, they were recovered at um, um, not closely related. So for conclusions, we recognize 13 species in the complex Davus pentaloris that are morphologically and genetically distinguishable and are concurrent with the geographical distribution. The morphological differences, the male sexual structures provide enough evidence about the species limits. However, by integrating different types of evidence, the species hypothesis become more robust. And well, the core marker was efficient to establish species boundaries between the species complex of Davus pentalaris. However, because we include few specimens from some populations, we need to include more specimens to see if this marker is also useful for when we include a large number of specimens. And also, when it is necessary to include the morphology of the missing and confirmed candidate species to test if the morphological and molecular evidence are congruent between them. And DNA barcoding could be very useful for future efforts in conservation of this species complex. Also because we want to, after the species were described, maybe we can propose that they could be protected by Mexican laws. Also, the practices and description of new species based on hobby material or its series should be banned in the future for increasing the stability and systematics of Theraphosidae. So I want to thank many people, and I want to mention that this study was dedicated to the memory of Norman Platnik because his enormous contribution to arachnology. And I want to thank also all the different creators of the different collections that let me to revise the many specimens.